Okay, I'm going to take the opportunity here to review some very basic genetics and the mechanisms of genetic change for a course in human origins. The building blocks of life, human DNA, it ultimately comes down to nucleotides and the way nucleotides are arranged and the information that they provide proteins. So here, really briefly, what we need to know for our particular course is there are four nucleotides in DNA: adenine, thymine, cytosine, and guanine. In RNA, we have a replacement of thymine for uracil. So in the human genome, there are about 3 billion base pairs, and they follow a particular rule identified by Chargaff. So this is therefore Chargaff's rule. When referring to nucleotides, we can use notations. Where is my, there we go, notations. So adenine is represented with an A, thymine represented by a T, cytosine represented by a C, and of course guanine represented by a G. And the way that these nucleotides pair, adenine always pairs with thymine, and thymine only pairs with adenine. Cytosine pairs with guanine, and guanine pairs with cytosine. So in a DNA strand, for example, we might get something like A, T, C, G, A, T, C, G, A, C. I don't know. So we get pairing. So when our polymerase, a type of acid, is replicating DNA, it's going to always make sure that the pair for A in a particular strand is complemented by the thymine T. So T is complemented by the A according to the rule, C with G, G with C, A with T, T with A, C with G, G with C, A with T, and C with G, and this is replicated in that strand. So each of these can be considered sister strands, and each of these nucleotides, they're read in sentences, and they are constructed in what's referred to as a codon. So codons are read in threes. So here we have we don't have quite enough nucleotides to have ourselves 4 codons, but there are 3 codons present. So as explained, these pairs, these triplets of nucleotides in our codons, they ultimately produce the instructions to build proteins that are responsible for creating our phenotypes or our physical features. So when it comes to the expression of our gene, our nucleotides, they produce what we refer to as a genotype. So this is all the genetic material that we have in our genotype. It is expressed as something that we refer to as our phenotype. This is our physical features. Think about pheno-physical. Nice little connection there. So our nucleotides, these sentences, they're transcribed, they're translated in our body, and it expresses those genes, whether or not we're tall, whether or not we're short, if we have brown hair, if we have blonde hair, if we have blue eyes or brown eyes. Now, a gene is a particular segment on a chromosome. And it's not proper to talk about a brown eye gene or a blue eye gene, and actually in a number of different ways. Number one, notoriously, this is an issue because eye color is coded by a number of genes. And for demonstrative purposes, we usually talk about eye color because it's something that we can relate to. So keep in mind that eye color is actually fairly complex, but we frequently hear things like they have a gene for brown eyes. She has a gene for blue eyes. He has a gene for green eyes. I wonder what gene what my child will have and the color of eyes they will have. Well, everybody has this particular gene that's going to be responsible for coding eye pigment. It doesn't change. It's always going to be found in the same location. So a section of DNA, that's called a gene. And when we get a biochemical difference on this particular location in this gene, that's going to produce the difference between brown eyes and blue eyes, or even coding for particular diseases such as cystic fibrosis, and we'll talk about that in a minute. So then we can say basically genes code for traits. Alleles are responsible for the variation in the traits that any species possesses. And as Charles Darwin pointed out, variation is necessary for any species rather than use, and here's kind of anthropology in the way that we think about it. Variation, people have used this to create barriers between individuals, use it to oppress individuals and to create perceived differences and to place people into discrete units. With humans, there are a few things that we can place us into discrete units, for example, whether or not we have a hitchhiker's thumb or not. But when we look at a lot of our traits, for example, skin pigment, we see continuous variation, and we group ourselves according to Klein's. This variation, ultimately, it's something that needs to be celebrated because this produced the human species. The variation that we see, this is our success as a species. Anyhow, moving on to the next slide. Right, I'm going to begin with a correction, really, because we're we're not talking about gene expression. We're talking about allele expression, or what types of traits will be expressed. And that's okay. We can make mistakes. We can correct ourselves along the way. Um, it sounded a little bit like Bob Ross. Anyhow, we're gonna on the right hand side we see a set of P's. We'll use P's because we're talking about Mendelian genetics, and Mendel apparently liked peas. I'm assuming beans as well. And we have a number of different traits or alleles associated with the skin on these P's. Again, if we're thinking about it, there's going to be a particular gene that's going to be responsible for a general trait, such as skin texture. And in this case, we have an allele that's responsible for saying smooth skin texture, and another allele that's responsible for wrinkled skin texture. So we have this nice smooth surface, and we have this nice wrinkled surface. And here we're going to apply really an unbiased perspective in every situation, unless it's providing a disadvantage. Both of these are fine. It's variation. We see variation in all populations as expressed. So what Mendel had discovered was some alleles are dominant and they cancel out recessive traits. So for example, if we look in the case of, so here we'll have, say, parent 1, and on the top we'll have parent 2. So for example, if parent 1 passes on a recessive trait for skin texture, producing wrinkled skin, and mom passes on a dominant trait, round, we're going to have a situation where the offspring, so this section of our Punnett square, where the offspring has a dominant allele R and a recessive allele R. Now this situation here where we have a dominant and a recessive, so two different traits, dominant plus recessive, this is heterozygous. And in this particular situation, the dominant trait is going to cancel out the recessive trait, and what's going to be expressed is an offspring with a round, smooth surface. All right, and remember, just because what's being expressed phenotypically is round and smooth, that doesn't mean that the other traits disappear. That can be passed on in another generation, because what we're seeing, round, smooth, this is in the phenotype. And the genotype here, we still have this recessive trait. It's possessed. In the other situation that we see here, I'll use the Punnett square up at the top. If mom passes on a dominant gene and dad passes on a dominant gene, the offspring will have two dominant traits, and therefore the individual is what we're going to refer to as homozygous. So here we have dominant traits, let's say times 2. So that's one particular situation where we'll have a dominant trait expressed. Now a recessive trait, it's only going to be expressed in this homozygous state. So we have a recessive trait times 2. So that's going to equal wrinkled. So that will be expressed in the phenotype. And with respect to the dominant traits, when we have 2 of those, in this case, we're going to get the smooth. All right, let's move on to our next slide. In the next set of slides, I want to talk about an example from humans, other mammals as well, but I want to talk about sweating, because sweating is something that is very unique to the human species, and it helps us understand the circumstances within which we evolved, and also why are we so sweaty and sometimes so stinky. And this is an evolutionary thing. It's about natural selection. So with respect to our course material, like I said, there's going to be a few more details here than we actually require on a general level, but for individuals who are planning to go into anthropology or another area, you might take away a little bit more. Okay, so as pointed out, we sweat. And over on the right-hand side, we got a great picture. I love photography that catches those moments. There's so much being said here. Exhaustion, just endurance. Wow, look at that drop of sweat. Why do we sweat? And how is this connected to evolution? So when we think about this, we should always be thinking in terms of other species, not that we're better evolved than other species, that we're more evolved or that there's a hierarchy. But in a lot of situations, yes, we are better evolved for certain types of environments. And that's true for all species. Humans, we are, like I said, very sweaty. Our sweat gland density is about 10 times greater than any other mammal. And mammals do have a problem, all mammals in general. We have an issue with overheating. And when we overheat, this messes with our brains. So sweating and temperature regulation or thermal regulation, and we will talk about this later on. This is a really important key to human evolution. It helps us understand the environment within which we evolved and some of the adaptations that were selected for naturally. So the density of our sweat glands, it's a result of an adaptation living on the savanna, and what we know about our evolution and what paleoenvironmentologists tell us. During the Miocene, there was a drying event, and a lot of forests were lost in the eastern portion of Africa, really producing the environment and the geography that we see currently. So imagine humans, our ancestors, are evolving in an environment that is threatening to the human species or any mammal. Now, we can adapt in a variety of different ways. So, for example, behaviorally, lions, take a lion, for example, behavioral adaptation to the heat of the savanna when the sun is at its highest point, and in around that high point when UV radiation is quite high, they take to the shade. And no one is getting a lion out of the shade. You know, behaviorally, they can do that because they have the power, the strength as an apical predator here, the top of the apex. They're going to do what they want. Other species really don't have that advantage to push them out. So changes like density in sweat glands, that's going to help the body cool. Now, there's a particular gene, and like I said, this isn't going to be tested, the particular gene, but we have something that's referred to as EN1, and it codes for a protein that causes our skin cells to form sweat glands. And the amount of sweat, this is going to be controlled by another gene. So there's a long evolutionary process taking place here where a number of pieces of our genetic code are starting to fit together over time and being selected for naturally. Now, I'm going to introduce a term here with respect to our sweat gland density and this EN1 coding for proteins that cause skin cells to form sweat glands. We're going to add a word here. This is a result of mutation. And remember, mutation refers to these random changes that take place, and if they help a species acquire more resources and therefore reproduce more, another key term, that particular mutation will continue to persist in the genotype and be expressed because it is creating more individuals that have that particular feature. It's beneficial. All right, let's take a look on the at the image on the right-hand side of the page, and I'll go through a few notations here for individuals who are interested in starting to develop some knowledge in genetics. Like I said, the amount of sweat is produced by another gene. It's referred to as the CFTR gene. We see that right here. And then underneath we see some numbers and letters. What does that mean? Chromosomes notation represented here, 7. So this is telling us that the CFTR gene is found on chromosome 7 on the long arm, Q, position 31.2 on that banding. Okay, so P and Q and R, there's no R. So the short arm, this is referred to as P, and our long arms on chromosomes are referred to as Q. And while we're here, we'll say that our short arm P and our long arm Q, they're connected with something in the center, referred to as a centromere. And there's little tails. Let's add those happy little tails in there. These happy little tails, they're called telomeres. Now, the amount of sweat controlled by this CFTR gene. It's responsible for telling a protein to make water, essentially, in tissue. And sometimes there's going to be mutations that are going to produce less water, and other mutations that are going to produce more water. And depending upon the situation, one might be beneficial, one may not. In the case that I'm thinking of here, I'm thinking about a mutation that's responsible for causing cystic fibrosis. So a mutation in the CFTR gene, this particular location, is causing cystic fibrosis, which is a recessive trait. In other words, the only way that it is being expressed is when we have, and here we'll use the notation from the gene. So a capital C will represent normal production of gene, water, and a small c producing an excess of water in tissue. So in the case where we have a capital C, we have the normal production of water in tissue. In a case where we have a capital C and a small c, in this heterozygous form, the dominant trait is going to cancel out the recessive trait. And in the situation where we have the small c and the small c, this homozygous form, this recessive trait will be expressed. Cystic fibrosis is a disease that produces an excess of mucus and is life-threatening. Now, cystic fibrosis carriers, it's also been found that they have an advantage against or for cholera. So in the form where we have a heterozygous expression, or expression in our genotype, so it'll be capital C, small c. There is protection against cholera, and this is also found among certain populations as well. So that clinal type of distribution that we had talked about when we had debunked the idea of biological races, but we also wanted to point out that there are clearly differences in human populations based upon the geographic regions that we have individually adapted to. So we refer to those populations as clines. Now, chromosomes are read, like I said, in a variety of different ways. They're different sizes. They contain different amounts of information. They do different things. For example, the SLC24A5 gene, this is found on chromosome 15. And it's responsible for differences in skin pigment. And this is important to understand because it helps us understand the ways within which we have evolved individually, the variation that has emerged among humans, and how it has provided populations an advantage in the geographic regions that they've eventually occupied. Here, with respect to chromosome 2, the second largest chromosome, this is particularly interesting from an evolutionary perspective, and we need to talk about it because it is a new chromosome and it helps us understand kind of the divergence that our earlier species took. Now, as one of the things I've explained is some people, they think that evolution is can be best understood or is understood in a anagenic model. So in other words, we have our point here where we have a common ancestor or our last ancestor, and it leads to a new species, and then that one leads to a new species, and that one leads to a new species. And I read this stuff on social media, for example. Read something on Facebook, and somebody said, well, if we came from chimpanzees, how come chimpanzees are still here? Well, they're clearly using the anagenic model to understand that, which is number one problem. And there's so many issues with that particular statement because humans did not come from chimpanzees. We share a common ancestor. So if we go down here, we share a common ancestor, and there was a speciation event that sent us on two different directions. Now, this doesn't mean that the anagenic model isn't accurate either. Okay, it needs to be understood within the cladogenic model. So the bottom one is the cladogenic model that helps us understand speciation. I'll fix my D there. And on top we have the anagenic model, which does not explain how humans emerged over time. So within this anagenic model, and if we say down on the bottom we have the human line, we do want to say that there is evolution taking place on this line, and it's best understood, I think, perhaps as twists and curves along this line of human evolution where we have variation, but we're not diverging from this line far enough, any particular group of humans, that would lead to a new species. So this helps us understand variation within, sorry, that's small, within a species. And remember that humans are 99.9% genetically identical. All right, so this chromosome, this new chromosome, what are we talking about? Okay, so let's start off. How many chromosomes do humans have? We currently have 46, and at one point of time, our ancestors had 48. How do we know this? Okay, so review, I'll give you your points here on your PowerPoint. We've said it's the second largest chromosome. And how did we get it? We've got a fusion of two chromosomes that continue to remain separate in other apes. And remember, humans are a member of the great ape family. We're all hominids. Now, what ended up happening, and we continue to refer to the chromosomes, for example, in chimpanzees as 2A and 2B, whereas we have the human chromosome is just referred to as 2. So here we see 2A and 2B. They're short arms, petite. There was a mutation event where those two short arms engaged in a head-to-head fusion at a point in our evolution. It's placed at about 900,000 years before the present. So that would mean that Homo erectus, okay, so that's the species we have at that time, there was a difference in some of the genetics of those species. Some Homo erectus would have 48 chromosomes, top corner, and some would have 46 chromosomes. And this would produce, if they were interbreeding these individuals with different chromosomes, ultimately this would create a problem. There wouldn't be enough genetic information for the offspring to go on and be fertile. Okay, so here, very simply, this is kind of the magic of my PowerPoint, Chris Angel stuff here, not. So we have our chromosomes that are fusing. There we go, and they fuse at the centromere, and we have an X chromosome. Now, in the case of the human chromosome 2, there's this mutation as pointed out. So we have an ancestral chromosome 2A, ancestral chromosome 2B, and they're going to do their thing here. There you go. They've mutated. There was a head-to-head fusion, and you're thinking, I know you're thinking, Wow, that was amazing. Awesome PowerPoint. Yes, that is good as it gets. Okay, tongue in cheek there. I try to make it interesting for my students. Anyhow, what happened here was a deletion of the long arm on ancestral chromosome 2A and ancestral chromosome 2B, leaving humans with only the P arm of the past two chromosomes. And if this is true, I'm going to say here, if this is true, what we should see really, I would think, is some vestigial um, evidence for that, right? So when I say vestigial, we say at one point of time, how do we know that humans had a tail? Well, we have a vestigial tail. We refer to that as the coccyx. Um, and here we see evidence of a vestigial centromere. Now you're thinking, well, Prof, Pop, you drew that in. That's not proof at all. And I agree, this is demonstrative. So let's take a look at a magnification of chromosome 2 and see if we can pinpoint two centromeres. Because if we do have two centromeres on chromosome 2, and you can't have two centers, that means that one of them um, is vestigial and gives us the information that we need to know. So again, yeah, no, I know this isn't the magnified electron microscope one slide. Human chromosome 2, HSA2. So labeling this, we have the centromere as such. This is the current centromere. And up here we have P, the short arm, and down here we have Q, the new long arm, which is actually a past short arm. Anyhow, moving down this chromosome, we have an inverted telomeric repeat at 2. Okay, so this is second chromosome, long arm at position 13. So that information is provided just under the new centromere in HSA2. And if we progress down a little bit further, we have a vestigial centromere at 2Q, position 21.2. And as pointed out, this tells us that something significant had happened in human evolution that really set us on a different path from our ancestors. And the other thing that I'll point out here is that 900,000 years ago, we also suspect that there was a serious bottleneck event, and I'll talk about that just before, at least the concept of a bottleneck event, a bottleneck effect, before we finish up this slide. And here we have the magnified version of actual chromosomes, and in particular the chromosome 2. We're looking at this one right here, and we can identify that it is chromosome 2 because it has two centromeres, enlarged centromere 1 and centromere 2. So that shouldn't be there pre. We don't see this with chimpanzees or gorillas on chromosome 2. So pre 900,000 years ago, that wouldn't have existed, and that's just again an example of a mutation that takes place. Now I'm seeing another one here, two chromosomes, and we can go through this as well. Down here I'm seeing chromosome 2, two centromeres. I see two centromeres here, and we can go through that as well to identify our chromosomes. Okay, so our mechanisms of evolutionary change that we are going to be interested in human origins, and this also applies for any species. Number one is the process of mutation, and new mutations produce new features. Mutation isn't necessarily bad. In some cases, yes, a mutation is not good. In some cases, a mutation does absolutely nothing. We went through an experiment with marbles and colored gels, and we saw that some mutations made no difference with respect to camouflage, whereas other mutations, for example, red gel mutation, made the green marbles appear dark, more similar to the black marbles. Therefore, they had camouflage. So that would be a mutation that would remain within the population because it's going to lead those particular individuals within that group to reproduce at a higher rate. So there are four types of mutations. So, for example, number one, we get something that's referred to as a substitution mutation. So nucleotides are substituted. And here, if I get my black marker, SLC24A5A111T, a substitution here. A and T produces a different in skin pigmentation. And we'll talk more about mutations in our later modules. There's something that's referred to as insertion, where information is inserted in. And think of DNA strand. It really, if we could take DNA and stretch it out, it would go from I'm told, I've read, from the human genome would go from the Earth and back 8,000 times. And it would look a whole lot like a big snotty mess. DNA can twist, and when it twists and curls, you get issues with the way nucleotides are aligning and the way that they are being replicated. We get something that's referred to as deletion and something that's referred to as a frame shift mutation. Now, one of the ways that I had explained this in class, and I had students go through an experiment where instead of using A, C, T, and G, we constructed a sentence based on codons. The big bad fly had one eye. Well, that's spelled wrong. Let's fix that. Had one eye. Okay, so we have our codons and we have a sentence that is being read. Now, if there was a particular deletion here, say, I don't know, I asked students to use a random, the Google random number picker, say that there was a deletion in the Y. When the sister strand is being repeated, the big, so the codons are being repeated, bad fly, now there's a space here. So H moves over, and we complete rest of the DNA strand, and there's a problem with reading this. The big bad fla adoni yi. So there's a mutation here, and I'm not sure how it's going to affect the species, but anyhow, this is one particular mutation with illustrating a deletion and a frame shift. Now, mutations, they are expressed on coding DNA, and other mutations. I'll add in another term here, so two more terms. We get SNPs, or single nucleotide polymorphisms. Oh, I had enough space, great. SNP is referred to as a single nucleotide polymorphism. This is a type of mutation. The other types of mutations that we're interested in occurs on non-coding DNA, referred to as UEPs. A UEP is a unique event polymorphism. talk about that later on. So anyhow, our mutations, they can be expressed and that they provide an advantage to the species. For example, sweating more, therefore cooling more, therefore being able to exploit the warmer periods of the day on the savanna, leading to reproductive success. Then they will be selected for naturally. So we get this idea that Charles Darwin had offered natural selection. And as I pointed out, a lot of people think that Charles Darwin was responsible for the idea survival of the fittest. However, that's not true. Another individual by the name of Herbert Spencer was responsible for that phrase and published it a number of years later. Charles Darwin, yes, he did begin to use the term survival of the fittest in his book Origin of Species in later editions. So natural selection is the process through which everything operates. If the mutation provides a reproductive advantage, more resources, more babies, more genetic material, mutated genetic material being passed on, and it stays within the population. Now, with respect to staying in the population, so like I said, we have that line that I had drawn with respect to human evolution. I'll go down to the bottom. The human species, we are 99.9% genetically identical, but that doesn't mean there isn't variation. So we get some variation on this line, accounting for that 0.1%. We get other variation on this line, other individuals, and that those differences that we see. Okay, so we're all on the same line. I'll use one more color here. We're all on the same line. These differences, the mutations, they are adaptations. I'll write that in. They're adaptations that are ultimately directed by geography, the location where we live, our environment. And we refer to the differences that we see. This produces what we call clines, not races. So gene flow, I'll go to one more color here. Can I get a different color? I don't know. Something that's going to stand out, maybe this one. When we get, that doesn't really stand out, but either way you can see it. When we get individuals here, to use the vernacular, Netflix and chilling coitus, having sex, that allows genes to be shared within the human species. So gene flow refers to all of the copulation that humans engage in between populations. We share those genetics, and that binds us as a species rather than leading to speciation. Okay, so random genetic drift. This will lead to increased diversity, and if populations are separated, if they are isolated from each other, it will lead to speciation. The other term that we talked about, and we'll talk about in greater detail, is something referred to as a bottleneck event. And humans, we've gone through a number of bottleneck events. For example, 900,000 years ago, there was a bottleneck event, and about 70,000 years ago, there was a bottleneck event which reduced human populations to approximately 1,280. Yes, you heard correct. Go to the top. 1,280 breeding individuals, approximately 70,000 years ago. And that's a subject for a later lecture. So this reduces genetic diversity, and think about it just as it sounds. Take a bunch of marbles with variation in a population, stick it into a bottle, a soda pop bottle, give it a shake, turn it upside down really quickly, and see how many individuals get through that opening. What's happening is that's a crisis, a very quick event that's jerked populations, and only a couple get through. If you do the same experiment, shake it up, and then kind of move it slowly, we're going to get a few more marbles to come through, and that's going to say that the event, yes, it reduced variation, but not all variation. Now, there's another term here, the founder effect, and when individuals leave a particular population, migrate out, and perhaps they're carrying certain genes. And the other thing that's not here, that really should be alongside of natural selection, is also sexual selection. This is something that Darwin talked about as well as a force in change. And remember, he Origins of Species didn't talk about evolution. He didn't use those words. He was talking about descent with modification. But sexual selection, discussed later on, this is something that, again, comes down to perhaps preference, to perhaps discrimination. New feature emerges, and animals, they do. They discriminate. We see that with birds. Certain dogs, I see my dog, she'll discriminate against other dogs. I don't know why. Some dogs she loves. Most dogs she does not like. She thinks she's a predator. She gets down really low and starts hunting them, and she's not big. It's weird what's going through her mind. Either way, we get a founder effect when individuals that are carrying, say, a new trait or a different trait leave a population, and they carry that trait to their new location. And in doing so, we start to see that trait become the dominant trait with them. So, for example, in the Isles, we see areas where red hair is dominant, and that could be a result of the founder effect. In the United States of America, for example, certain groups of individuals, Amish that migrated out of Europe, they carried a particular gene, and we see a high prevalence of a sixth finger. So that would be, again, responsible in part by something called the founder effect. Okay, that's where we're going to leave it. That kind of wraps up a lot of the things that we've talked about. Hope you have a great day. Take care.